Now we're going to do this a little more, we're going to make this a little more complicated so that we can run data analysis on it. So what I'd like you to do is take do a second variable here, AAX is going to be equal to cbind. Can everybody read this so I don't need to read it out aloud? Big enough? Oh, here, let me make. Why don't I do this? Why don't I let people just type these in, and then after you've typed in this function, as you get to the end of each line, go ahead and type in, for instance, AAS once you've entered that, and just make sure you can visualize what it looks like. I find the most helpful thing is just to constantly print out those variables so that you can see what you're doing. If you just type in the code blindly, eventually you lose the forest for the trees. If anyone has any errors or questions, please raise your hand. I'd be happy to. So when you get down to this plot function, I've made this much more complicated than it needs to be. You can just plot AAS2 by AAS1 and then close parentheses. This just adds some labels and some decoration to the plot. Okay. Why is yours black? Yeah, that's pretty cool. Sorry, I need to give people a yes, sir. I'll be. What happened to Mandy? You. Oh, your test is capital. You capitalized it there, but not there. Okay, means morning message, notification, non-factors ignored, X unexpected, comma. Here you didn't pick the second set of parentheses around test. So that's what I'm saying with it. It'll give you, it really tells you where your trouble is, and then you just got to sort of look around that area to see if something doesn't. Yeah. Okay. You know, I think I remember getting a warning message when I ran this last night on the means. But it was I couldn't replicate it. I don't know what the. I don't think it's that big a deal. When you got this, did you get the summary? This is the more important thing. The summary there. It sort of shows you. Let me run that real quick for everybody, and then we can. I'll come back to this so people can continue to type in, but let's just go over as a group real quick, sort of this basic result. Yeah, I can probably get rid of this mean statement. I don't think it's that useful, but. Okay, so I just ran the same code. You all did? Yeah, it gave me an error here. Okay, what this is telling me is that it is significant. Right? So we're testing in the ANOVA. AOV is an ANOVA test, and it turns out that there is a significant difference between the levels. And so what do those levels look like? Well, it's this AAS. This is where you bound everything together into a single analysis. And this is just saying that at level 2, here is some means. At level 1, here is some means. At level 0, here is some means that you randomly generated. Now, when you plotted this, for those of you who've gotten to this, and my plot's a little more frilly, because it's not found. Means. Oh, I think this means needs to be capitalized. But what you can see is you can see the difference here, and it should have given you your equation. Just like I was showing here, you get some equation. Okay? And you're testing the mean separation between these. Now the point is, so we have significance, so we know that this locus is significant, and if we can get this equation, that tells us the effect. The effect of going from little a to the heterozygote to the big A, big A. Okay. Any questions on that, other than why my code isn't working when? Sorry. Do people want to? People want to continue to enter some of this, or did everyone get it mostly figured out? Should be testing means capital. I think that's the. Still a problem. Yeah, I don't use this model.tables ever. This was something I put together a long, long time ago, and so I don't remember what it's pulling up here. Oh, could be these guys. Could be those quotes for mine. Yeah, it's those quotes for mine. Pull that up. Image effects means. Okay, so. That happens afterwards. It's not find. Oops. It's not finding something it needs to find. Ah, lowercase again. Okay. Nope. Paste. To. Oh, it still saved it. Yeah. Okay. There we go. Okay, so it calculated the tables of means and estimated the effect. So in my simulation here, the effect was 4.95, when what we actually simulated was a difference of 5. So now, I think I have it in this code that it plots. Oh, this is going to be the same problem. So for effects, I need to get rid of that. Yep. Lowercase e, yep. There we go. Thank you. So now when I do this. Okay, so now we got the line and we got the effects of going from the lower to the heterozygote, the rounded effects, difference between big A, and little a, and it was significant. Sorry about all the problems here. That the quotes between PowerPoint and R always give me problems. Okay, any questions? I'm sorry, that wasn't very clear. Any questions on getting that code entered in? What we did there was just generate random numbers for the 3 different classes. We bound them all together. We ran the ANOVA on them. We tested them for their means, and then we plotted them. I mean, that was the gist of what I wanted to show real briefly. Now, for our QTL, you won't do any of that. All that functionality is already built in. But what I wanted to remind people is that in this case, there's a difference between the effect and the significance. So in a QTL mapping study, the significance, or whether or not we detect a QTL, is going to be determined both by the difference in the means of the classes, so the difference between big A, and little a, but it's also going to be dependent upon the number of individuals you have. So if you only have 10 individuals, you're not going to be able to map any QTL. Replicates and environments are going to somewhat affect that. I mean, we're essentially going to analyze within an environment. But the more replicates you have, what does that increase our ability to do? Having more reps. So we'll reduce the experimental error. Well, actually, our error will be larger, but by having more replicates, we'll be more accurate in our final estimation of that value. Yeah. So you're going to have more accurate phenotyping data, theoretically, from having more replicates. The variance within each class. So if we changed our standard deviation from 3 in this example to something like 9, then it's going to spread these points out quite a bit. I mean, I can do that real quick, and there's probably a trick in RStudio to rerun everything I already typed in. Let's just see what happens if we change the standard deviation on this experiment to 9. And this code's going to be wrong because of this. Okay, so now what we see is it's no longer significant. Okay, the difference between homozygous AA, big A, and homozygous little a is the same. All we did was we changed the standard deviation and the spread of the individual observations. So that's consistent with having additional effects in our population or less accurate phenotyping data. And we see we still have a slope of our line, and there's still maybe even a 10 difference, 9.07. So the difference is the same. It's just no longer significant because there's too many other things going on, too many other sources of variation. Now I'll give you a little example of that here. Make the slide big. So remember, we're testing for the significance of different markers. So this is one marker. If you're doing a genome-wide scan, you might have thousands of markers. And so the class here, there's 50 observations for homozygous dominant, 50 observations for homozygous recessive, 100 observations for the heterozygote. Their difference is 110 to 100 in phenotype value, and the standard deviation is 3 here. And so we estimate about the effect is about 10, and the significance is very high. Now if we change that so our standard deviation is greater, our effect is similar, but our significance is much lower. This is with more observations than you had. So you were only doing, I think, 50 individuals you were simulating, and here we're simulating 200. Now if we change to a dominant model, or codominant model, what we see is our significance is pretty similar and our effect is pretty similar, because we're only using an additive model and analysis here. And then if we reduce our number of observations, we still get the same effect estimate, but we've really dropped our significance. Okay. So again, this is just simulating what we think might be happening in reality. So for the examples that we're going to use over the next few days, we're going to be, I think, using primarily real data that was collected in the field. But you don't know whether or not there's an effect. You might get a significant effect, but you don't actually know if there is one in there. Whereas what we're doing here is we're just simulating some of this data, so we know that there should be a difference between this category and this category, although in some cases we don't do a very good job of detecting it. We detect it with low significance. And so if we make our effect size smaller, so the difference between here is we go from 100 to 110, 100 to 110, 100 to 110, and here we go from 100 to 102. So the difference in our effect is lower. We estimate that well, but look at the significance here. Significance here is 0.002. Significance here is e to the minus 16. So the bigger our QTL is, the bigger our QTL effect is, the more likely we're going to detect it with higher significance. So once we have this lower effect and we add more standard deviation into our individuals, we lose our ability to detect that. Questions? I'm going to skip over some of this, or I guess the rest of it. So the point of the QTL mapping exercises we're going to do is not necessarily single marker analysis, but I just wanted to give you a little tutorial with R on how you might look at single marker analysis in a simple way. The next step would be to go to interval mapping, and then eventually composite interval mapping, Well, we're using the genetic map to test for effects both at markers, which are these little carets here, and between markers. So I don't have anything else I was planning on covering. I think that covered the main introduction. But the other thing I wanted to save time for is any type of discussion, answering basic questions, comments, concerns, criticisms. Ace. Hmm? Good question. I've never used a rename function. I'm almost positive there is one. But for most of the stuff we generally do, the data sets are small enough that it can handle probably 1 million objects without any, it can handle thousands of objects. So it's just, for me, it's always easier to rename the object whenever I manipulate it or do something different, and then have them both in memory. But I'm sure there's a rename function. I just don't know it. Somebody else here probably does, though. Or you could rename it and then delete the old one with the RM in parentheses. Good question. Other questions from people on either R or QTL mapping? Nothing? Okay. Well, oh, Sandra. That's a good question. That's a very, that's sort of a technical question. The question is, do we use composite interval or single marker analysis? So I'm actually a big proponent in that there's no one right way to do analysis. I think it's really important. I think a lot of us sometimes have this idea that there's this one right way to do analysis, and my analysis method is better than yours. But generally, that's going to apply if you have a computer simulation and you know the answer. What I'm really looking at as a plant breeder is to try and find something that I'm looking for and have the most confidence to do that. And what I've found over the years is the way I have the most confidence to do that is to run every possible analysis method every possible way and look for consistency of results. So if I run single marker analysis and then I run interval mapping and I run composite interval mapping and I found the same QTL in all three analyses, I feel much better than if I only find it in one. Same thing if I find it in every environment versus if I find it in one. Okay, those are two different problems, but the same thing. So what you're trying to do is build up the body of evidence to actually move on with that QTL or with that marker. So if you look at previous studies, for instance, that'll also help you. A lot of it gets down to what's your end goal. Is your goal to clone a gene? I mean, because if it's to clone a gene, you really want to be sure that you're following the right QTL. If it's to do marker-assisted selection and it's cheaper just to, do a cycle of mass and then remap the QTL, maybe that makes more sense. So a lot of it's going to depend on your goal. But we have single marker, we have interval mapping, we have composite interval mapping, and then we have some Bayesian methods. And all those are going to give you slightly different results. So for my graduate level class, which there's, I don't know, maybe 6 or 7 people in here who have taken, we simulate QTL, and then we run through all three methods. And what most people find is, maybe only 10% of the QTL, even a huge population with a lot of markers, are found. But if you run all three analysis methods, you're more likely to find more of those QTL. So each method has its own little abilities to find things. In genomic selection, there's this method called random tree, I think, prediction, or random forest prediction. And so it uses a lot of different models, and then it looks for syntheses between them. So I just feel like we're doing the same thing. We're running a lot of different models and we're doing synthesis. We're just doing it on a much slower scale than some of these automated methods. But yeah, good question. Theoretically, composite neural mapping is supposed to be the most powerful and best, and generally it is, but that's not always the case. You mean it's conservative? It makes too big of a QTL? Too liberal. Okay. So so just on a research note here, we have the population I was mentioning, 1,400 lines, 107,000 markers. We actually find for one of our 4 Mendelian traits, even with single marker analysis, it actually predicts the QTL in the wrong position. So QTL, again, are statistical depictions of where loci are, not biological. So it's not, just because you find a QTL doesn't mean it's real and doesn't mean it's in the right place. I don't know if I can ever emphasize that possibly enough for people. But it is a statistical detection. It is one of our best techniques for detecting loci, but it's not absolute. Yes, sir. Well, what's your goal? Or are you talking about me personally? Me personally, the best QTL I ever found was the one I was most excited about was in sorghum, and then I switched crops, so I didn't get to do much on that. And to date, still nobody has been able to they've tried, but they haven't been able to follow it up. I think because it's a gene that's not in the reference genome. That's my hypothesis. On the other hand, in maize, most of the QTL studies I've done, I haven't really wanted to follow up, but there's an association study we recently did, and so for that, what we're following up with is with another population. Because if you look at what the, in a linkage mapping population, you might have 500 or 5,000 genes under a QTL. So even gene expression isn't going to be that helpful. And you still have this question of what time point and what tissue are you looking at for that gene expression. So I think the first thing to do, personally, is do another QTL mapping study with a different population, different set of environments, different genetic mapping markers, and see if you detect the same thing. And if you do, and you have that confidence, then it's worthwhile pursuing. But just one QTL study, I don't think, is enough to ever pursue a single locus. Gene expression can be helpful, but almost all the examples I've seen, I've never actually seen anybody find anything under a QTL based on gene expression. But yet, very few people follow up on cloning genes in crops. And then gene expression is also a technique for cloning a gene. And for a lot of people, especially in A&M, I think the marker-assisted selection is much more important, and you don't need to get to the gene. For marker-assisted selection, a QTL of this size is perfectly fine, because you're not going to have any more recombination than that in your following population. So what's your goal? What's your population? All these things play into it. Well, I mean, I wouldn't. So the way you would do marker-assisted selection with the Caspar marker is you would probably have the 2 flanking markers at least of the QTL region, and from Sandra's comment, maybe you would make them even a little bigger, and then the primary marker. But this is, we spent about 3 weeks going over this in my graduate level class, so I'm kind of answering your question, but I don't think I'm really covering everything that needs to be covered. Yeah. Anything else? Well, there's a lot of caveats there. So I would say that, in what I'm showing you here, this is the major QTL and this is the minor QTL in this population and in this environment with this set of analysis and techniques. Well, I can't develop expression of that QTL in my population because there may be 500. If you're talking about gene expression, there may be 500 genes under here. So which gene is the QTL? Is causal to QTL? Or is it the sum across all 500? Is it 500 genes with very small effects that sum up to one QTL? Or is there a major gene in there? We don't know. Well, what's your goal? Is your goal to clone the gene, or is your goal to use it for breeding? Oh, so if you're trying to find if you're trying to find QTL for improving your cultivar, then yeah, you would probably take the one with the largest effect, and you develop some marker system like Caspar or some array-based system to follow those markers in subsequent generations. But I'm a firm believer in Mark Cooper's approach, and Mark Cooper will be here next week. He came up with a paper that we read in my class called Mapping as You Go. And the whole idea is that you do 1 or 2 cycles of selection, and either this QTL becomes fixed, or the genetic background becomes different, so this QTL's expression may not be as useful. So it's not like you do 1 QTL study and you're done. It's you do 1 QTL study, you select based on that, and then you do another QTL study on that population, you select based on that. So you're continuing updating the model of what the best QTL is, based on your new environments and your new populations. Yes. Anyone else? Okay. And it's important to remember that Carl Broman, when you guys see him tomorrow, his expertise is really in biostatistics, and his primary focus is in mouse and human and animal systems. But he's generally very gracious in trying to work with plant scientists as well. But we all have different sort of limitations and needs. So one of the things I think the main focus is on the software and the analysis. That's why I wanted to give you an opportunity. If you have any confusion about QTL mapping itself, it might be a better use of time to either discuss that with your colleagues or myself or other people or Carl. But again, tomorrow the main focus is going to be, I think, on learning as software. The main focus, not the only focus. Okay. Well, thank you guys all for your time.